Welcome back, people. Looks like chapter, what's this, 13, isn't it? No, 12. I can't remember if that's kind of already. No, 13. He's on variation, alleles, and mutations. So, looking at karyotypes, describing chromosomes, determining sex, and then how mutations occur. We're going to talk about silent missense and nonsense, how we might detect those, what mutations might occur, and how do we go about repairing mutation. So, I think we've discussed karyotype before. It's one of those things you need to be aware of, and we can easily karyotype people. Just need a sample of your DNA. This is a person here who is clearly human and clearly male. And I can tell they're male, once I get this back up, by the fact that they've got one X chromosome and one Y chromosome. And 22 pairs suggest then to me that this is a human male. And we can get that sample from amniocentesis by using all sorts of different techniques to sample tissue. Sperm, mucus, skin, hair, blood, get some white blood cells out of your blood tissue. All sorts of different ways. And then, of course, we pair them together. I think that's the next slide, isn't it? No, we'll go back a minute. So this is pair 1. Pair 1 is longest. They're slightly longer than pair 2, pair 3, pair 4, etc. And pair 22, the smallest. We then line them up on their centromeres. So that one is essentially metacentric. That centromere is essentially in the middle. This one here is slightly to one side, so it's sub-metacentric. These ones here are much better. These are definite sub-metacentrics. That centromere is away from the centre. And then, let's see if we can find a nice little agrocentric one. Well, no, humans don't have one. These are sort of agrocentric, they're not really. An agrocentric one will have the centromere right at the top. And clearly from that diagram, humans don't have those. They haven't asked you that sort of question on the exam for a long time, but just, a bit of nomenclature doesn't hurt. So, they can be homozygous they can be homologous or they can be non-homologous. So an homologous pair of chromosomes are identical, they hold the same genes. A non-homologous pair would be non-identical, they'd have different genetic material on them. And I'll just describe this stuff here about how we go about naming them, but that's not really that important. Interesting enough, of course, sex determination is one of those things that comes up. And the famous old question was always about Henry VIII killing all his wives because they couldn't give him sons. But of course, the problem was his own DNA. The male in mammals determines the sex of the child because they pass on the Y chromosome in half of their sperm. The female can only pass on X. So it's the male's job, if you like, to determine sex. In birds and reptiles, and some reptiles and amphibians, it's different. And we use a different system called Z and W here, where the mother is ZW, and she passes on her W, and that causes femaleness. Whereas the father just passes on his Z. They often, if they do ask you any questions about this, and they probably won't anymore, but they used to, this is a situation where they like to use ZW because it reverses it all, and it makes you think it through. And they can catch you out. We love our examiners. So what's really important to us is the idea of mutation. Essentially, for every gene that we contain, there was once only one variant. So at some point in time, some great progenitor, way back, what's Richard Dawkins call them? I feel like Timmy uses for those ancestors of importance. Processor, procenter, something like that. At some point in time, they were identical to each other. But as we mutate, we create variations of each gene. And variations can be quite beneficial. So we look at that around our classroom, and you've all got different eye colour, different hair colour, different skin tone, your different heights, your different body shapes. This is all normal. And that's why we get so upset about some of the stuff happens in media about body shape and things. We are all human. We share a common humanity, but that humanity is really quite diverse. And that diversity is found in the alleles. So the more alleles available for a particular gene, the greater diversity for that particular gene. So when our sheep showing team are breeding their sheep, they're looking to get animals together that haven't been from the same family, because they'll bring different alleles, and that means the offspring will be stronger. And a population is strongest when it has greater genetic mutation. So with our quolls, when we're breeding our eastern quolls, we quite deliberately try and make sure that animals that are breeding at Woodleigh have never been together before. So we get animals from Dunkeld, we get animals from Rothwell, animals coming from Tasmania, they come down from Mulligan's Flat and Canberra, and we rotate all these animals around to try and create situations where we get diverse allele frequency. And that diversity, that variation within the genome, within the population, is the strength of a population. So mutations are useful, not always bad. And we've got here a little situation with different wing types in fruit flies. This one's not much chopped anyway, is it? Look at those wings, they're not going to fly anywhere. Vestigal, no longer working really. Rudimentary, no pro-fly. Normal would have been the original, the wild type, and all these are variations on that. We've talked before about the fact that dark skin, dark hair, dark eye colour is all what the wild type of humans would have been as we arose in equatorial Africa. We talk about point mutations. This occurs at a single point on a chromosome in the gene sequence. And they can be one of three things. They can be silent, so the mutation's there, we've changed the actual nucleotide sequence, but it codes the same protein. It codes the same amino acid. Can't see it. It may not code exactly for the same amino acid, but a closely related amino acid, and again, we don't see the change. It doesn't make a phenotypic effect, something we can see. So it's not deleterious. It's not advantageous either. It's just done nothing. It can create missense. So we've got a change in amino acid sequence, and that could produce a new functioning version of that gene. And missense probably accounts for all those variations we have. Or they produce nonsense, and the protein either finishes quickly, early, comes too early, or doesn't stop at all and just keeps on going, or the protein structure that's created is such rubbish with so with no potentially useful active site that it's of no value at all, and they're nonsense. So how does this happen? Well, we can do substitution. So we have our template strand here, TAC-ATA-CAT. That will give me methionine, tyrosine, and valine as the three amino acids. And if we mutate that by substituting, I've substituted a G here for that A, that changes this particular amino acid to his, whatever his is. New protein, possibly. So that could be missense. It may well be silent. It may not make any change to the amino acid, to the protein. We can delete one. So here I've taken this C out. And so that moves that A to there, that T to here, and then we get something else coming in. I've added A just for the sake of it, making it easy. And we now have two tyrosines in a row. Again, we've changed the amino acid. Silent, more likely missense, possibly nonsense, we don't know. But it's a change, it's a mutation in the nucleotide sequence and a change in the amino acid sequence. We can insert one. So again, I think I picked the same spot I did too. So there's C. We've thrown a G in first, GCA. This is now arginine. Again, we've changed the amino acid sequence. What does that do? Silent? Missense? Nonsense? Don't know. So we talk about mutations occurring in the germline or in the somatic cells. So somatic cells, my body cells, who cares? It may cause my death. There may be mutations which cause me to develop cancers which I can't cure or don't cure in time. Or they just, they're changed. Big deal. I can't do anything with them. I can't pass them on, which is the important part. Or they can be germline. So changes, mutations in the development of my sperm will cause variation which I can pass on. That may be a good variation, it may be a bad variation. Only my son or daughter will find out. But those germline ones are the ones we're most interested in because they get passed down. And they're the most useful ones because, again, they get passed down. We get new things developing, and that's important. How do we go about detecting mutations? Well, of course, we can sequence nucleotides for all our genes. So we actually physically take a sequence of DNA that we know the gene exists in and sequence it and compare it to others using electrophoresis and breaking up with restriction enzymes and DNA probes, all the stuff we talked about in previous chapters. Easy thing to do, and people do it all the time now, of course, because it's part of disease management, especially in cancers. What causes these changes? Well, they're what we call mutagens, agents which cause a mutation, and they're not that rare. Fever. How often do we get fever associated with diseases? Mutagen. Exposure to radiation, so sun, which we're all well aware of the need to be careful in the sunshine. So sun is a constant cause of mutation at the epithelial layer, but often deeper. X-rays, of course, are, again, essentially electromagnetic radiation like the sun, but penetrate much deeper. And, of course, carcinogens, things that cause cancer, cancers being mutations to our DNA anyway, carcinogens, of course, are also mutagens. And so we're thinking of things like nicotine and chemicals like DDT and so many other things, asbestos, that sort of stuff. But, beautifully, our cells can repair themselves. So there are enzymes whose name now won't come to me because I want it to, or I'll check it out before we have class. These enzymes actually run along looking for damage. So in mitosis and meiosis, they're trying to make sure that the damage doesn't get passed on. Because a lot of that mutation can occur in misreading during mitosis as well. So we have little mistakes made. But generally they're fixed because there is a repair system in place just in case that happens. If it doesn't happen, again, in mitosis, that can lead to apoptosis. So the cell doesn't get to develop and pass that mistake on. But it has to be picked up. I think that's the end, isn't it? Yeah. That's the end of chapter, whatever it was, 13. And I might pause there because it's quite cold out here in Carl's bedroom. And I'll load this up for you. And come back tomorrow and make some more. See you tomorrow.